Hello, my name is Alan Jacob, and today we'll be talking about the propagation of action potentials. So what is an action potential? The body is completely encompassed by the nervous system, which is a network of nerves and neurons that help the body communicate within itself. And so like if your arm wants to do something or your brain tells your arm to do that through the nervous system. And so your nervous system is effectively like the information highway of the body. And an action potential is kind of like a car on that highway carrying the information. And so an action potential is the way that like neurons transport these electrical signals throughout the body. And so how does an action potential start? Well, an action potential requires a stimulus to start. And a stimulus just means something that is going to trigger the start of an action potential. And so, but like it can't be any stimulus. It has to be something that meets a certain threshold. And so that's kind of like if you were trying to push a car by yourself, you might not be strong enough to get that car rolling. And that'd be like a stimulus that hasn't met the threshold because the stimulus is being received because like it's similar to how you're pushing that car, but it's not meeting the threshold. You're not strong enough to get that car rolling. But now let's say you get a group of your friends and everybody's helping out, then together you might be strong enough to get that car rolling, and that would be like a stimulus that does meet that threshold because it would generate an action potential. And before I talk about how action potential travels, I want to go over the parts of a neuron real quick. And so this yellow stuff that you see is kind of like fingers. Those are known as dendrites. And that's the part of a neuron that receives the stimulus and receives these electrical impulses. And if the stimulus is strong enough to surpass that threshold and go and create this action potential, it will form an action potential at the beginning of what looks like this tail-looking thing, this red thing called the axon. It'll start an action potential and send it down the axon, and the synaptic terminal is where it'll be passed on to its destination or passed off to another neuron in the chain. So as far as propagation itself goes, so like I said, an action potential is passed from a neuron to the next through the axon, and the axon works a lot like a wire. So if you are charging your phone, you might plug in the charger into the wall and then plug it into your phone so the electricity can go from the wall and the outlet to your phone to charge it. So similar to that, the axon takes these electrical impulses and these action potentials from the axon hill clock, the beginning of this axon, beginning of this tail, and send it down the axon, effectively like a wire, to your phone, or in the case of the axon, to a neighboring neuron or the destination. What you have to remember is that this axon is like a one-way road. When you're traveling down the axon, you can't turn around and go back the way you came from. A good way of thinking about this is Lightning McQueen on the track. So if you think of the track as the axon and the pathway that the action potential travels, and that Lightning McQueen as the action potential, as he travels on the track and passes a certain point, think about spikes coming up, and these spikes prevent him from going back through that point that he just crossed. But eventually when he makes it around that track, he's gonna have to pass that point again, right? Similar to that, after an axon propagates an action potential, once it sends that action potential down the axon, it has to use that area again to help transport the next action potential that's coming. So likewise, the spikes go up just for a moment after Lightning McQueen passes it, and then when by the time he comes back around, the spikes have gone down, so now he can cross it again. So the spikes restrict him from moving backward, but they don't limit him from moving forward. And that's a lot like the axon because the action potentials move forward and they don't come back, they don't go the other way. So now at a high school level, an action potential is the method of communication that the body uses through the transport of electrical signals. And it's not actually a molecule that's carrying this information. So it's not like DNA where there's actually something carrying all this information. it's actually a rapid rise and fall in membrane potential, which is the difference in charge across the membrane between the extracellular and intracellular. And so the resting potential is the equilibrium of the outside and the inside charges and what that potential is. So in a neuron, it sits at about negative 70 millivolts, and a stimulus that exceeds that threshold of negative 55 millivolts will create an action potential, generating a depolarization effect. And during depolarization, sodium is entering the axon cell body, and the axon is typically negatively charged on the inside and positively charged on the outside, which is what gives it the negative membrane potential at resting. And so the increase of sodium, which is positively charged, creates a local positive with inside the axon, and that sends the membrane potential positive and puts it at about a positive 40 at this peak. And then when that happens, once it peaks out, the body wants to return to equilibrium of the negative 70. And so to account for all these positive sodium ions that are entering, that just entered the axon cell body, during repolarization, positively charged potassium ions are released to bring that membrane potential back to that negative 70 value. So during the depolarization, like I said, sodium channels are activated and sodium enters the axon as shown here in 1. And then that depolarization activates the nearby sodium channels. And so this sodium influx that made this area positive is now going to activate the sodium channels in this adjacent area. That's going to increase the sodium levels. And so as sodium enters the cell, it increases the effect of depolarization, increasing the number of adjacent sodium channels that are activated, increasing the concentration of sodium. So it's a positive feedback loop. But once the action potential, the depolarization has occurred for the action potential, like I said before, it goes to the repolarization stage, which is visible here in 2 as the potassium is being released back out to bring the inside back to that negative. And during this repolarization stage, the sodium channels in that region are inactivated, and so they cannot transport sodium into the axon, and therefore it cannot initiate that depolarization effect that creates an action potential. And so during that repolarization, there cannot be an action potential that is transported through that region. But as that action potential moves on and that repolarization also moves on to that next region, this region that was originally being repolarized has returned to that resting state. And so now in that resting state, it can entertain a new action potential. And this is what allows action potentials to be sent out in high frequencies, because you don't have to wait till an action potential reaches all the way to the end of the axon. You just have to wait until a certain region is no longer in its refractory period or repolarization stage. But as far as travel goes, there's 2 main ways that they travel. Myelinated, because there's 2 types of neurons, myelinated and unmyelinated. Myelinated neurons are neurons that have myelin wrapped around the axon. And there's areas where there's actually no myelin, and these are known as the nodes of Ranvier. And so in myelinated neurons, the action potential that's created, which is shown here, it could, instead of traveling through the axon and through these myelinated areas, it jumps over the myelinated area into the nodes of Ranvier. And so if instead of going through the entire axon, it's effectively skipping down the axon, which makes it faster. The unmyelinated neuron, however, does not have this myelin wrap or the nodes of Ranvier. And so when action potentials are started and propagated through the axon of an unmyelinated neuron, rather than skipping down the axon as in the myelinated neuron, it it's kind of walking down the axon, and it goes through the entirety of the axon, which is continuous conduction as opposed to the myelinated neurons that have saltatory conduction. And so, and they propagate until they get to the destination. And now at a college level, so an action potential is the method of communication within the body. And it's highly dependent on the rapid rise and fall of membrane potentials in the axon. And with a resting membrane potential about -70 and a threshold of a -55, stimulus has to bring membrane potential above that -55 value to create an action potential and instigate the depolarization effect. And during depolarization, there's an influx of sodium ions through the sodium channels, and after the depolarization is achieved, it's followed by repolarization, in which the membrane voltage, the membrane potential is brought back down to the -70 value, which is the resting state. And so during this repolarization stage, the membrane potential is brought back down through the release of potassium ions. And so when it's propagating, the sodium entering its channel creates an electric field that travels through the hydrophobic part of the membrane, so the tails, and it activates the neighboring sodium channels. And so what happens here is, as this area is depolarized, the sodium entering through these channels that would be right here is creating an electric field that travels down the membrane. And so this electric field, as it reaches the sodium channels that are in this region that's currently not depolarized, this electric field activates the sodium channels found in this region, increasing the depolarization of that region. And as this depolarization continues down, the area that was just depolarized begins repolarizing to reestablish that equilibrium of value of -70 millivolts. And during that time, it experiences a refractory period in which the sodium channels are inactivated and cannot entertain an action potential because it can't depolarize. But after the repolarization is complete, it returns to the resting state of -70 millivolts. And at that point, it can entertain an action potential again. And so something I've mentioned on is the importance of the membrane potential and how crucial it is to determining action potential generation and propagation. And to make sure that the membrane potential is where it needs to be, we have leak channels for sodium and potassium to help regulate the membrane potential and fine tune it and ensure that action potential generation and propagation occurs flawlessly. And there are two types. There's myelinated and unmyelinated neurons, and they the action potential travels down them differently. So in myelinated neurons, there's a myelin wrap around the axon, and these areas that are covered in myelin have low concentrations of sodium channels. But the nodes of Ranvier in between the myelinated areas have a very high concentration of sodium channels. So when this electric field is being spread down the membrane, when it reaches the myelinated area, there's a very low concentration of sodium channels that can get activated. So the myelinated area doesn't experience a very large depolarizing effect. But when that electric field reaches the nodes of Ranvier, the high concentration of sodium channels found there are activated. And so it creates this effect of skipping, where the action potentials are skipping over the myelinated areas because there's not enough channels to create a depolarization effect there, but there are enough in the nodes of Ranvier to depolarize there. And so the action potential is effectively jumping over the myelinated areas and traveling through the nodes of Ranvier through what is known as saltatory conduction. Whereas in unmyelinated neurons, the concentration of sodium channels is about level or equal throughout the axon due to the lack of myelin and nodes of Ranvier. And so rather than jumping from node to node, it travels down the entirety of the axon as the electric field that travels through the membrane activates adjacent sodium channels throughout the entirety of the axon, causing unmyelinated neurons to follow continuous conduction. And so as these propagate, they get to the end of the neuron and they repeat the cycle in the next neuron, and they keep going until they reach the destination.